So today we're getting into the last section of evolution, and we're talking about the process of speciation. Speciation is the formation of a new species, so how different organisms that were once part of the same species become different separate species. Okay, so as new species evolve, populations become reproductively isolated from one another. So reproductive isolation is going to be one of the major hallmarks of speciation. That's how we define whether or not they are still the same species. So reproductive isolation is when members of two populations cannot interbreed and produce fertile offspring. So fertile offspring is an important one there because obviously we know that donkeys and horses are different species, but their ability to breed with one another does not make them the same species because the offspring, which is a mule, is sterile. So you get a sterile offspring, so that negates the whole thing there. They are still reproductively isolated. Behavioral isolation is an isolating mechanism which occurs when two populations are capable of interbreeding but have differences in courtship rituals or other types of behaviors. So you see this on planet Earth with the birds of paradise. Okay, the birds of paradise, they do a different dance, they do a display, they set up a little stage, they go through all these steps to try and attract a mate. But if they're different species or if the mating rituals are separate from one another or differentiated from one another, that's when they become behaviorally isolated. So their dance, their display separates them, and they will no longer interbreed with something that another bird that they possibly could have, but that is going to keep them separate. Okay. Geographic isolation is two populations are separated by a geographic barrier, such as rivers, mountains, or bodies of water. Now, this is an example of allopatric speciation. Allopatric speciation. Make sure you make a note. Allopatric speciation is when they are in two separate ranges. So the ranges do not interact with one another. Okay, the two ranges, the species are geographically divided. Now, temporal isolation is when they occur at different times of the year. So this happens big in plants. We all know that there are spring and fall allergies. All right, spring allergies are starting now. Fall allergies we get at the end of summer. But we know that because of this, we know that the allergies are caused by pollen, and pollen is the male reproductive portion of a plant. And because of this, we know that they would be temporally isolated from one another. They're not reproducing in the same time. Now, temporal isolation and behavioral isolation can be examples of what is called sympatric speciation, where two species live in the same range. They're in the same exact area, but because of different behaviors or different reproductive times, they're isolated from reproducing together. Okay, so sympatric, same area, but they don't reproduce. Allopatric, different areas, they don't reproduce. Those two terms are very important. Make sure you make note of them. They will be on the quiz. Okay, so we see right here Galapagos finches. All right, Darwin's most famous example. So we see that we have a wide variety of beaks found in these finches. But if you see, you can see that they have different food sources. So those different food sources are going to be very important to which niche they fill. Okay, and you see they have different habitats, they have different food sources. So this is going to allow them to reduce competition. Okay, they occupy a different niche. Once they occupy a different niche, they start to no longer interbreed because of behaviors, and they become different species. So how the Galapagos finches speciated. A few finches travel from South America to one of the islands. There they survive and reproduce. Separation of population. Some birds from species A cross to a second island. Two populations no longer share a gene pool. So because of this speciation, because they're separate, again, they're in different places, so this would be allopatric. They don't share a gene pool. Okay? The gene pool doesn't work anymore. All right? It doesn't flow back and forth. And the example to think about with a gene pool is it's actually an example from a literal pool. I talked about it during division, cell division. And one of the things that we have is when I was a kid, we went to this hotel that had a indoor-outdoor swimming pool. Okay, part of it was inside, part of it was outside. You could swim back and forth through a little, it wasn't even a tunnel. You could walk back and forth through it, but it was a little doorway. And what would happen is in the wintertime, you could see there were tracks where they would slide it down and block it out. So when you imagine you put a bunch of pool toys in the indoor pool, a bunch of pool toys in the outdoor pool. Now in the summertime when they're both open, those genes, those pool toys, the pool toys represent genes, flow back and forth between the two sections. But if you close them off, now the gene pools are no longer shared. Okay, now that's an important thing to think about when we talk about founder's effect, because if all of a sudden all of one type of genes are found in one when you close that door, now you have an allele frequency that's based off of random chance, not based off of natural selection. Okay, so that literal pool, think about it as a pool full of pool toys or a pool full of genes, when they could flow inside and outside, is important. Now, sympatric speciation, if they were in the same area and they could still interbreed, that's what these finches would be undergoing. But now, since they're separated on two different islands, this is allopatric speciation. All right. Changes in gene pool, seed size on the second island favors birds with larger beaks. Now it's favoring a different set of genes, more selection, naturally selecting for them. And now if they cross back, they're reproductively isolated because their behaviors are different. They're fulfilling different niches. They're eating a different food source. They're not really interacting with one another anymore. And competition. As species A and B compete for seeds in the first island, they continue to evolve. All right, they're always trying to fill a different niche. Competition is not good for species. Now, obviously, it selects for the strongest. We know competition is important, but they're always trying to minimize competition. So this process continues, leading to all 13 species on the Galapagos. So in terms of the early Earth, we talked about this a little bit during our presentations, but the heterotroph hypothesis is a proposed explanation of how life arose and evolved on Earth. So the first things on Earth were heterotrophic, the first living things, and they obtained organic nutrients. So they would have undergone fermentation. Fermentation, we know, releases carbon dioxide. Because there was no free oxygen at the time, that carbon dioxide built up and allowed for autotrophs to evolve. So because you had autotrophs, okay, so here is the simple organic fermentation, and here is autotrophs, now can evolve because there's CO2 in the atmosphere. Autotrophic activity was carried out, and what do autotrophs release? Plants release oxygen. So that allowed for the first aerobic organisms to come about. Now, we're going to switch gears a little bit here, and we're going to talk about some of the evidences for evolution. So unfortunately, this is covering, but it's a good picture, and it's showing homologous structures. Now, they have different organisms here. We have a human, a cat, a whale, and a bat, and they're very different, but notice how they have all the same bones. So those same bones are all going to come together. They have the same origin, same bones, and that is a very, very important thing to think about when we're dealing with these, that they are homologous, all right? Same, homologous, all right? Remember we talked about homozygous and heterozygous, homo meaning same, so these structures have the same origins and we have the same bones, okay? If you think about it, our leg too, our femur would be analogous to our humerus, tibia and fibula to the radius and ulna, tarsals to the carpals, metatarsals to the metacarpals, and toes to the fingers. All right? Now, if we look here, comparative embryology, these are different embryos. The early embryos of all of these different animals look so similar. They're all vertebrates, all right? So they all have very similar characteristics to one another. So the combination, the fact that these embryos look so similar shows a common ancestry, okay, common ancestry. Then comparative cytology, all living things are made up of cells, mitochondria, ribosomes, cell membranes, structurally and functionally similar in most eukaryotes, okay. Prokaryotes still have ribosomes. They obviously don't have mitochondria because mitochondria are believed to have been prokaryotes at one time. And biochemistry, DNA, RNA, and proteins, the central dogma of biology applies across the board. Okay, The central dogma applies across the board in this case.